But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell.